So the next speaker, uh, Professor Ichiro Takahashi. Um, so I said, he's last almost 40 years, almost 4 decades, right? Almost 4 decades. He's working in photosynthesis related topics, and his research is very focused on algal photosynthesis, especially chlamydomonas and hali. So, and also as I said, um, his lab is established with a lot of equipment with full of biochemistry and molecular biology instruments, where he may be focused. I mean, last another
You need
even
some
about 10,
for
11,
YouTube.
12 talks. In that he may cover from
That is YouTube.
from biogenesis to the functional aspects,
It's a microphone.
as well as the biochemical aspects, and also one talk will be also on structural aspects of photosystem one. So I think tomorrow we'll be going to have more of formal introduction in front of Vice Chancellor. Again, I would like to thank Chiro for coming here and sharing your knowledge with the participants, with the students. So please go ahead.
Microphone is on. You can hear me? Okay. Thank you very much. Very kind introduction, and I'm very glad and honored to be here again in Hyderabad. This is my I don't know, fifth visit. Since I first visit to Hyderabad, I became a very fan of Indian food, spicy food, and very very nice. And okay, so first I introduce myself. When I started my this research on the photosynthesis was just a bachelor's thesis. It was 1979.
Yeah,
So at that time I was working with thermophilic cyanobacteria.
that's what I said. 4 decades.
4 decades, okay, yeah, more than
More than.
longer than. And also, at that time, why I used thermophilic cyanobacteria was the reason was the proteins of thermophilic cyanobacteria is very stable. So you can isolate the proteins or protein complexes that's usually fragile. We can isolate and characterize these complexes or proteins more easily. But I felt that at that time this thermophilic cyanobacteria was not amenable to molecular analysis like recombinant DNA or transformation, and at that time no genetic information was available. And then much after I got a PhD degree, I worked a little bit, a few years in the plants, the spinach. But later I decided to use the green alga, the Chlamydomonas reinhardii. I just started to use Chlamydomonas in 1989, and that time just as I did some the work in the University of Geneva in Switzerland to learn the molecular biology of Chlamydomonas. And then at that time, just they developed the selectable marker for chloroplast transformation. So then I joined this project and wanted to know to knock out or modify the photosynthetic genes encoded by a chloroplast genes using Chlamydomonas. So that is my starting point of using the research on photosynthesis using Chlamydomonas. because genetic information and chloroplast and nuclear transformation, although I haven't experienced the mitochondrial transformation, that is nothing to do with photosynthesis, I think. So I did the nuclear and chloroplast transformation using chlamydomonas. So that is a very powerful, this alga is very powerful organism and as a model organism. And there are also many the mutants deficient in photosynthesis. So there are many resources we can use. So that's why I'm, how to change this? This is pointer?
You can change the slides.
No pointer.
You can change the slides.
Only change
Yeah.
slide.
Yeah.
Okay. So here I'm going to molecular mechanism of chloroplast biogenesis, in particularly the more introductory talk, and then in my other lectures, I will going to give you later more specific topics. I will talk. So, of course, we know that plants and algae contain the chloroplast, but this was so. This is just a summary of the 3 genetic system in the Chlamydomonas reinhardii to show the pointer is here. Okay. Let's sit down. Okay. Okay. So this is Chlamydomonas.
Excuse me. Can I use it?
Pointer.
It's not coming. Maybe it's better. Maybe it's not getting. It's better. You can
This one? Okay. Of course, the nuclear genome has the large size, about 110 megabase pair, and the chloroplast genome has only 205 kilobase pair, much smaller. And mitochondrial genome in Chlamydomonas is even smaller, 15.8 kilobase pair. And genes encoded in this genome is 585, about 20,000 genes in nuclear genome. But in the chloroplast genome contain only 99 genes, and mitochondria even smaller, 37. So this gene in the chloroplast and the mitochondria also contain some genes encoding RNA for use for gene expression, ribosome RNA gene, and also transfer RNA genes, and also in the case of chloroplast, it contain RNA polymerase or the translation, some of the ribosome RNA, and also some ribosome proteins. Not all. Some are encoded by nuclear gene. Some are encoded by chloroplast genes. So this is a kind of a minimal size. And chromosome and copy number, the nuclear genome has 17 chromosomes. This is haploid, okay, not diploid. And chloroplast genome, Chlamydomonas has 1 chloroplast. Unlike plant cells has many, tens or more than 100 chloroplasts, but in Chlamydomonas it's only 1. And there are many mitochondrial genome, but it's about 130 mitochondria. Each mitochondria has 130 copy number of mitochondrial DNA. So, and usually we grow the Chlamydomonas in haploid. So if you have a mutation in the nuclear genes, if it has a phenotype, you can observe the phenotype because it's a haploid. If the cell is diploid, one of the pair of the genes, if that is wild type, you don't see phenotype, right? So this is very easy to screen the mutant that is deficient in your specific phenotype of your interest. So inheritance is a Mendelian nuclear gene in Mendelian law tetrad in the segregation. And in the chloroplast, mutation is inherited in maternal. Maternal, it's we distinguish this male or female, but it's designated as a mating type. This is mating type plus and mating type minus. Okay, the mating type plus is inherited from mating type plus cells if you genetically cross. But in the mitochondria, the inheritance was paternal. So from the mating type minus cells, its genetic information is transferred to the progeny. And if you introduce foreign genes from outside the cells, the gene DNA is integrated into the genome randomly, in the case of nuclear genome. But in the chloroplast genome and the mitochondrial genome, introduced DNA is integrated by homologous recombination. So you can, if you have homologous sequences in the introduced the DNA that are targeted to the right place of the DNA. So you can change specifically the genes of your interest in the case of the chloroplast and the mitochondria. That is very convenient. This one. Oh, no. Keyboard.
Yeah.
Here. Okay. So this is just summarize a history of the, not the whole history of the chloroplast, history of the discovery of chloroplast or characterization of chloroplast. I just took from some the source book of Chlamydomonas source book, the recently published in 2023. So it's described that in 1905, the plant chromatophores results from the uptake of cyanobacterium by eukaryotic ancestral organisms. That is some prediction kind of things. The evidence was not shown at that time. And then 1962, these two scientists observed some fibers in the chloroplast using the similar green alga Chlamydomonas moybiusii. So probably they claim that this DNA in the chloroplast. And 1963, Saga and Sega and Isida, they actually purified and characterized CP chloroplast DNA from Chlamydomonas cerevisiae. So they found that the Chlamydomonas had the chloroplast contain their own specific DNA. And then later, 1967 and 1970, the endosymbiotic theory was further established. So Professor this time very detailed history was presented. And the next is that how the chloroplast DNA looks like. So since 1979, the chloroplast genes were being sequenced. This means that the specific gene was cloned and then sequenced, not entire chloroplast DNA. And then from 1980 to 80, restriction maps of chloroplast DNA were constructed, okay, by digesting different restriction enzyme or combination of two enzymes. So then you can connect a fragment. And then it was found that the chloroplast DNA is circular. It's not linear. So this characteristic of DNA structure is similar to bacterial DNA. So that is consistent with the chloroplast is derived from the bacteria, cyanobacteria. And since 1979, the mutations in the chloroplast gene encoding a photosynthetic apparatus were found. So that clearly shows that some genes in the chloroplast DNA encode photosynthetic protein. Okay, and since 1973, chloroplast protein synthesis was being characterized. So one of the way to identify which proteins are encoded by chloroplast and which are encoded by nuclear genes. In the case of the chloroplast proteins, encoded proteins, if you add the antibiotics of the spexulomycin or something like that, so then this specifically blocks the ribosome, bacteria type ribosomes. So it blocks the synthesis of chloroplast encoded proteins. And for example, if you use cyclohexamide, that blocks the translation of the by the cytosolic the ribosome. So then you can assign which by using a labeling experiment. Okay, so it becomes clear that some chloroplast proteins are encoded by chloroplast genes, and the others, or majority of the chloroplast proteins, are encoded by nuclear genes. So nuclear encoded proteins are synthesized in the cytosol and transported, translocated into chloroplast somehow. And then 2002, the first complete sequence of circular chloroplast genome from Chlamydomonas cerevisiae was reported. And then, of course, there are so many errors in the beginning, but in 2018, very much refined chloroplast DNA sequence was determined. So the characteristics of this Chlamydomonas chloroplast DNA is GC content is low and AT content is high. But compared to the nuclear genes, the GC content is very high in Chlamydomonas. That is the main characteristic. So if you do a sequence of chloroplast DNA, there are so many A and T, AAA, TTT, ATA, ATA, so it's somehow confuse us to determine the sequence. And also in the many years ago, the high GC content in nuclear gene is also makes more difficult to determine the nucleotide sequencing. So this is a genetic information. And 1988, Boynton et al. First reported a chloroplast transformation using a biolistic method. The ballistic mean the biological ballistic method is, maybe I will later in the other lectures, I will describe, talk about how to do a chloroplast transformation, but here I just want to say small metal particle, for example, tungsten particle, it's like about, the diameter is about 1 micrometer. We coat the surface of the particle with transforming vector, okay, and then deliver this small particle into the cells, into chloroplast by particle gun. Do you know that particle gun? So we deliver this particle at a speed of sound, for example, 300 meters or 400 meters per second. So this is so small, so we have to work in a deliver must be work in the vacuum. Otherwise, the air they accelerate the particle speed and then go into. So it may kill many cells, but at a low probability, some successfully the particle is delivered into the chloroplast. So maybe later I will talk about how to select the transformant of chloroplast, because this is very essential, because as I told you, efficiency, or it's this ballistic method kills many cells, and also many cells are not targeted by particle. So relatively small number of cells are successfully transformed. So we have to select, for example, 20 or 30 transformants out of the millions of cells. So this is very important to selection of the transformant. But maybe we discuss later other talks about this. And in 1990, the chloroplast transformation was successful in tobacco in the plants. So this is more complicated process, but it's possible now. Tobacco is the method is very much established. And 1991, Goldschmidt Claremont in the University of Geneva reported the AADA cassette. That is confer the spexomycin, streptomycin resistant phenotype. So this AAD code for an enzyme that inactivate spexomycin. That is, spexomycin blocks the chloroplast translation. Bacterial type ribosome it blocks. Sorry. Yeah, but if you inactivate spectromycin by ADA cassette expression, so then the cell can survive in the presence of antibiotic spectromycin. So only transformed the cells can survive, but non-transformed the cells dies. So this is very efficient selection method. Okay, so there are many types of the plastid. The chloroplast is one of the plastid. So etioplast, chromoplast, and chloroplast, leucoplast, amyloplast, elioplast, and proteinoplast. This is plant. But I'm working on the green alga. So green alga contain only, usually only chloroplast. And sometimes I will talk later, or sometimes other lectures, etioplast also generated in glandulars. This is a So others are not really found in Chlamydomonas, but you can find in the plants. Okay. For example, amyloplast is a potato, store the starch. Chloroplast you can find in fruits or color. And leucoplast is no colorless, but it store something. This is a kind of leucoplast. No color, but it store other products in the cells. Okay. So this is the interconversion of the plastid. Okay, I skip. And this is a typical structure of the chloroplast. This is a chloroplast from spinach C3 plant. So this is a nice shape of chloroplast. And then, as you know, the chloroplast is surrounded by 2 layers of membrane. That is called envelopes, and outer envelope and inner envelope, 2 layers. And also in a membrane system, thylakoid membranes are present, like here. This is starch. So this region is many layers. The thylakoid membrane is stacked, so it's called grana, grana stacks. So if you see this region, you can see many layers of the thylakoid membranes. And inside this envelope of thylakoid membrane is called luminal space. And this region outside the thylakoid membrane is stroma that contain many enzymes, soluble enzymes. And this is also a chloroplast from grown in from the high light condition, and this is the chloroplast from the spinach of low light grown. So if you look at the thylakoid membranes inside the chloroplast, you will see the difference. Okay, the chloroplast from high light grown the plants has less grana. Okay. So the number of the stacking is lower. And this is a low light grown spinach, and you can find easily the more stacks. So the reason is this is a strong light, so the light is not the limited for the photosynthesis, so the plant has sufficient light. So chloroplasts do not need to capture light more. Okay, and in the low light grown cells, this need to collect more light. So the chlorophylls, photosynthetic pigments like the chlorophylls are present in the thylakoid membrane. So if you want to collect more light, you need more thylakoid membrane. So then, to accommodate the large amount of membrane, you need to stack properly, right? If you have many clothes, if you just scattered on the floor, you have no space to live. But if you are nicely organized and in the furniture, you have more space. So that's the case. You need, if the chloroplasts need more thylakoid membrane, this is the best way to stack and organize. And this is a chloroplast from C4 plants, Z-maize, and mesophyll and bundle seed cells. So this contain PS1, PS2, this contain PS1, so that's also stacking structure is different. So chloroplast has a different characteristics in the structure. And also if you look at the green algae, for example, this is a Chlamydomonas. So single cell and single chloroplast. No, the plant has many chloroplasts in single cells, but Chlamydomonas has only one chloroplast. So it's large, about 40% of the volume of the cell is occupied by single chloroplast. So it's a lot. So if you look at the shape, maybe it's difficult. This is nuclear. This is schematic. This is nuclear. So chloroplast is like this, single chloroplast. No, it's quite different from the plant. Okay, because there's a nuclear here. The chloroplast is like this is a section. So like a cup. No, if you cut the cup, you look like this U-shaped. So cup-shaped chloroplast is a characteristic of the Chlamydomonas. Okay, and the cytochrome membranes you can find here, and in the Chlamydomonas has less stacking, as you can see. No, not a little bit stacking like here, but it's not like the spinach chloroplast. It's more like this, no, abundant cells of C4 plant. And this is a pyrenoid. It's contain the starch and also many enzymes, particularly Rubisco enzyme is concentrated here very much. But otherwise, the chloroplast is very this Chlamydomonas chloroplast is similar to the chloroplast of the plants. So this Chlamydomonas is also can be used for model organism for studying chloroplast instead of the plants, because Chlamydomonas can grow very easily. You don't need much space. It's cheaper, right? And also, what is important is Chlamydomonas can grow without photosynthesis if you add proper carbon source like acetate. This is very important because you can isolate the chloroplast photosynthesis deficient, the mutant deficient in photosynthesis easily. Okay. Okay, if you look at the chloroplast DNA of Chlamydomonas, this is like that. It's very okay, circular DNA. Okay, and in a double-strand DNA, so inner circle is the transcribed this direction, okay, anti-clockwise, and outer chain is transcribed is clockwise direction. Okay, so these genes in a square, it's colored, are the genes that is transcribed in a counter clock direction. Okay, and these genes are clockwise direction transcribed. Okay, and what is interesting is this is the genes classified. For example, this is green is photosystem 1 proteins, and dark green is photosystem 2, and blue is cytochrome B6F complex proteins, and yellow is ATP synthase. And this orange is Rubisco large subunit. Okay. These are related to photosynthesis, has function of photosynthesis. Okay, there are many genes, but for example, photosystem 1 forms a complex consisting of many subunits, and of course, some very important subunits are encoded by chloroplast genes, but others, mostly peripheral subunits, are encoded by nuclear genes. So this is a case also in photosystem 2, cytochrome B6F complex, and ATP synthase. It's a kind of mosaic. Okay, chloroplast encoded subunit and nuclear encoded subunit are combined to form a functional complex. And Rubisco also large subunit, but small subunit are encoded by nuclear genes. And also the others, this violet is the brown related to expression of chloroplast genes. Okay, this is RNA polymerase, and this is ribosomal proteins. And okay, and also RNA genes, genes encoding RNA, tRNA and ribosomal RNA genes are present. Because all the RNA that is necessary for gene expression is encoded by chloroplast genes. Because RNA cannot be translocated from the cytosol to chloroplast. So all RNA must be synthesized inside chloroplast. Okay. And also other genes, this is, how to say, pink. It doesn't look pink, but some are openly different that its function is not known, or FSH or YCF1 or these are other functions. Indirectly, some are indirectly related to photosynthesis, but the others are not related to photosynthesis. And there's an intron. The gray is intron. For example, this genes, PSBA, is encoding one of the reaction center subunit of photosystem 2. This gene has a intron. So green part is exon. So this is the intron. Okay. And also other interesting gene is here. CHLN and CHLB gene. This is related to chlorophyll synthesis, protochlorophyll reductase. So maybe I talk later, but this is a Chlamydomonas and some plants, or for example, needle plants or moss plants can synthesize the chlorophylls in the dark, but many other plants, land plants, cannot synthesize chlorophylls in the dark. So there's a one step of chlorophyll synthesis pathway that is light dependent. But this light dependent pathway in the Chlamydomonas has the two types of the enzyme that is dependent on light, and the other is independent of light. So Chlamydomonas, like as a moss or some pine needle plants, can synthesize chlorophylls even when they grow in the dark. Okay, so this is important things. Okay, and also interesting characteristic is this circular chloroplast DNA has this region, this thick line is inverted repeat. So exactly the same nucleotide sequence, a pair of nucleotide sequence are present in the chloroplast DNA, but direction is inverted. So, okay. The sequence is exactly the same, but the direction is inverted. So that's we call the inverted repeats. And this is a single copy region, okay, here. So this is large, and this is smaller, usually. Which is larger, I don't know. It depends on the organism, it changes. But larger section, single copy region is and also smaller single copy region, and also inverted repeats. That is the main structure of chloroplast DNA. But between this inverted repeat region, for example, this is large single copy region, and this is small single copy region, and this is inverted region, inverted repeats. Inverted repeats B, inverted repeats A, so direction is like this. But there's a flip-flop, the mechanism, so by a homologous recombination, this exchanges. So if that is the case, if it's exchanged, as you can see here, the direction of a short single copy region direction is changed, swapped. That happens, but doesn't affect any expression of the chloroplast genes. So we don't need to care about, if we're working on the function of gene or working on the gene expression. We don't need to care about, but this happens. Okay. And also, what is important in the chloroplast has activity of the homologous recombination, as I told before. Okay, so then Chlamydophis has single cell, a single chloroplast, and the plant has many chloroplasts. So in the case of plants, maybe there are many chloroplasts, so that you can, the cell can separate many chloroplasts, probably mostly equally. But in the case of Chlamydophis, it has only single chloroplast. So when the cell divides, chloroplast must, division of the chloroplast must be synchronized with the division of the cells. And also a DNA of chloroplast DNA in the chloroplast is 80 copies, but must be separated. By chance, if by chance, one of the chloroplasts, after the chloroplast is divided, one of the chloroplasts has no DNA and the others has all the DNA. That, if that happens, cell cannot survive without chloroplast DNA. So there's some mechanism how the chloroplast DNA is separated properly between both divided chloroplast. So this is about the experiment, how exactly the chloroplast DNA is separated. So before I explain this, chloroplast DNA is not present in the chloroplast, particularly the stroma region, as a naked DNA molecule. This is some the proteins bind associated with this chloroplast DNA to form a chloroplast nucleoid. It's not like a chromosome, but it's simple structure, but it's actually some proteins associated with chloroplast DNA. And then form some globule structure, maybe more than one chloroplast DNA nucleus associated to make a large forms. So this is a chloroplast nucleoid like this, globulous. And if you look at the confocal fluorescence microscopy, if you stain this, this is a yellow fluorescence proteins fused to one of the proteins that are associated with chloroplast DNA. You can assign by this fluorescence that where this chloroplast nucleoid are dispersed in the chloroplast. Okay, and then you can follow during a cell cycle, light cycle, and dark cycle, G2 and SM and G0 cycle. So this grow the cell, and then the DNA is synthesized, and then its cell division occur. So you can follow the proteins of the associated with chloroplast DNA is by following. You can see how the chloroplast nucleoid looks like. So this large nucleoid particle becomes smaller, and then somehow dispersed to form like as if a network-like structure. Okay, and then cells and the chloroplast are divided 2 times. So make 4 cells, daughter cells. So if this red is the chlorophyll fluorescence. Okay, so it's, if you look at the fluorescence, the microscope, chlorophyll fluorescence is just coming much strongly. Okay, you cannot avoid this, but this shows that where the chloroplast is. Then it's divided into 4, you can see, and then this globular chloroplast nucleoid is dispersed, and then after division, it also again the globular structure appears to grow and grows larger. This is the same thing, similar proteins, yellow fluorescence proteins. So this is how the 80 copies of the chloroplast DNA is divided into equally to after 2 division into 4 daughter cells. Okay. So what is important is this nucleosome doesn't move. If this protein, this chloroplast DNA is present in the stroma of the chloroplast, this should be moved, right? But it doesn't move, do not move. That means suggests that chloroplast nucleoid is probably associated with thyroid membranes. It's not free completely. That is the case, okay? So another interesting feature of the chloroplast DNA is oh, sorry, this is something. When we do the mating type plus and mating type minus cells, the crossed, genetical cross. Okay, then as I show in the second slide that chloroplast DNA or genetic information is inherited from mating type plus. It's not from mating type minus. So the chloroplast, this is not a cell division. This is a sexual cross. One of the chloroplast, the genetic information disappear. Okay, that is from the mating type minus, and one of the chloroplast information from the cell of the mating type plus only dominate in the after crossing. Okay, so this means that probably the after the fusion, the chloroplast DNA from the mating type minus should disappear or digested. Okay. That should happen, and actually this data show that happened. Okay, this is a chlorophylls, and this is a DNA staining the overlay. So this is after crossing like this in the beginning. 60 minutes, later, two cells fused. So this becomes the from haploid to the diploid, and two nuclei is still present. And this is a chloroplast DNA. After crossing, the still both from mating type minus and mating type plus chloroplast has the similar amount of the chloroplast DNA. Time is running out. Okay. So, but in the 90 minutes later, no, the chloroplast DNA from mating type minus disappear, digested probably, right? Just happens. So this is just the same experiment. Okay. Okay, so if you look at here, so DNA is disappeared. This was present but disappeared, okay? So this is a typical style of the chloroplast inheritance. I think I'm going to talk other lecture about similar the subject. So maybe the later part is I will talk later in later lectures. I'm going to talk about biogenesis more, but we have a still time to do this later lecture. Okay. Okay. So I stop here, and I'm happy to have the question or from you.
So I would say, it is not that questions, if you have a kind of interaction, it is always better interaction. Any questions you can ask, the main aim of this Indian conference to interact with each other and inculcate whatever the subject so you will be kind of interested later. So that is the very main note of this. So we can ask any kind of questions and interact with them. It is not like an exam. There is no exam here. So, you have any clarifications you'd like to discuss? Yes, first.
Sir, like you said that under the low light intensity, the size of the grana differs in this image. So does it affect the size of the chloroplast? Like if there will be more stacked grana, there will be larger size of chloroplast in both conditions, or the size of the chloroplast will be equal?
You're talking about this.
Very
so
good
in the
question. Yeah.
huh?
Very good question.
Sorry, the
Sir, will the size of the chloroplast vary according to the
size of chloroplast? That's I don't know exactly, but of course the color of the leaf changes, of course. Under low light condition, it's more stacked. The stacked region has, maybe we discussed other lectures. Major
The question is whether the size of
chlorophyll. Chlorophyll? The amount of chlorophyll.
chloroplast.
Chloroplast size is, I don't know exactly the size, because only the focus on the stacking. Size is, it depends on the organism, I think, but it's, I don't know, sorry. The size is maybe changeable, but there is no description of this aspect. Maybe that is a good point and to analyze, to check. You expect that if there are more stacks, the size of the chloroplast may increase. Maybe not. But I don't know exact data or the evidence the difference in size, because people focus on the stacking. This is stacking. The number is drastically changes, so people just focus on this aspect. But the size of chloroplast is, I don't know if it's very important for the function of photosynthesis. But, yeah. Other questions?
Yes, sir. Here,
Yeah.
I have one query.
Is your
As-
microphone?
Yeah, I think I'm audible. Yeah. You are telling about this chloroplast transformation. In that, particularly, NP plus should be the one which is going to transfer to the next generation. So ideally, using MT for chloroplast transformation, you should use always MT plus strain. That is the ideal one.
Sorry,
For as you said, only the MT plus strains is going to carry out the mutation to the next generation, right? In the chloroplast, whatever mutations you have made in MT plus.
mating type plus.
Yeah, mating type plus. So all the transformation will be done in mating type plus strain only.
Yeah, of course. That is basically yes. And after the chloroplast transformation, if you want to cross with some mutant that has, for example, the nuclear mutant, you can combine, chloroplast mutant and mutation and nuclear mutation together. But if you transform the chloroplast using a mating type minus, if you cross, you lose the phenotype. So of course, basically, almost all the case, we use a mating type for chloroplast transformation.
And added to this question, one more thing, Sensei, regarding the recombination. You said for transformation, the nuclear is random insertion.
Basically, recently we can tag
specific homologous also. So in that case, if you specifically, I think the procedure seems to be almost similar. What do you feel? Like chloroplast transformation versus a nuclear transformation. If you use CRISPR in this case.
Sure.
So I think the procedures might be almost similar. Based on the recombination, I think the site.
Yeah, of course, it's the vector is different, the method is different. I have not experienced CRISPR-Cas9, but it takes more time than random insertion. So usually we use nuclear, the mutant, as a host to transform of gene of your interest. Then the transformed the genes is randomly inserted in the nuclear genome, but it doesn't matter if it's expressed to complement the phenotype. So usually we use this method. But you need to screen because it's randomly inserted, so it may happen that second mutation may occur to if the transforming the vector inserted into some genes that may give you a new phenotype. But if that is not related to the growth of cell or photosynthesis, we don't care. But we need to isolate much more than one mutant transformant. And at least 3 transformants, we compare phenotype, and if they are the same, we can say that this is successful. And yeah, that's the case. So if you have the mutant, you can use this traditional nuclear transformation to complement, and also mutagenesis, cytodirected mutagenesis, or tag fusion. So if you have a nuclear mutant of genes of your interest, that's very powerful. But if you don't have, you need to use CRISPR-Cas9. So some people works very well, and some people don't do not. So that's sometimes risk. There is a risk of this method.